Hi class, this is Dr. O'Connor. Here we're going to talk about proteins and amino acid metabolism. So let's first start with the digestion of proteins. So remember, proteins can be denatured at low pHs. So the environment of the stomach is acidic. The pH is from 1 to 2. So therefore the proteins will be denatured in the acidic environment of the stomach. So pepsin is an enzyme that is used to catalyze the hydrolysis of some of the peptide bonds of the denatured protein. And so as these peptide bonds are hydrolyzed, what we end up with is polypeptides. The polypeptides then will enter the small intestine. Here the pH is about 7 to 8. And the proteases, these are also enzymes, will further hydrolyze the peptide bonds. I just want to go back up here for a minute, talk for a second about the enzyme pepsin. Most enzymes would be denatured at a low pH that we find in the stomach, but pepsin is definitely an exception to that rule. We're back here where the polypeptides enter the small intestine and they're further broken down. These peptide bonds are hydrolyzed even further. Basically what will happen is the amino acids will be freed from the intestinal lining. So after further hydrolysis, we end up with some amino acids and dipeptides in the small intestine. And then these free amino acids are transported across the intestinal lining and then absorbed directly into the bloodstream. Now, we refer to the amino acid pool, and this is the entire collection of free amino acids in the body. An adult will turn over about 300 grams of protein daily. And the amino acids will continually enter the pool from digestion and, of course, breakdown of old protein. Amino acids are continuously being removed for synthesis of nitrogen-containing biomolecules. Each of the 20 amino acids is degraded by means of its own unique pathway. So there's a different pathway for each amino acid. We're not going to talk about all of those here. So some of the nitrogen-containing biomolecules that are synthesized from the amino acids, one of them is nitrogen monoxide, and we refer to this as a chemical messenger. And it's a very interesting molecule. It is a free radical. Remember, free radicals are species that have unpaired electrons, so an odd number of electrons. So that makes it very, very reactive. It lowers blood pressure, it kills invading bacteria, and it's also known to enhance the memory. The nitrogen monoxide is synthesized in the linings of blood vessels and elsewhere from oxygen and the amino acid arginine. So in the blood vessels, the nitrogen monoxide will activate reactions in the smooth muscle cells, and this causes dilation and it results in a decrease in the blood pressure. So everyone's heard of nitroglycerin. What happens is the nitroglycerin releases nitrogen monoxide, and this is really important for treating angina, which is the pain experienced by people that have blocked arteries. Okay, so other biomolecules that are synthesized from amino acids, hormones, neurotransmitters. Oh, look at this. NAD and NADP, and also the heme molecule that we find in hemoglobin. So the end result of protein digestion is the hydrolysis of all the peptide bonds in the protein and the polypeptides and dipeptides in order to produce the amino acids. So let's go ahead and just talk a little bit about amino acid catabolism. The first step is the removal of the amino group, and this is where transamination comes in. This is the interchange of the amino group of an amino acid and the keto group of an alpha keto acid. Here's what we have. This is a enzyme catalyzed reaction. So here is amino acid 1. This is our alpha keto acid. That's because the alpha carbon has this carbonyl group. Okay, so the carbonyl group here is the carbonyl carbon is the alpha carbon. So obviously this is enzyme catalyzed, and here's what happens. This group here is transferred to the alpha keto acid. So this is the alpha keto acid with the nitrogen group bonding to what was the alpha carbon. So this is now another amino acid. Again, usually glutamate. And then we're left with an alpha keto acid. So when this group here is removed, then the double bond forms between carbon and oxygen, and that is converted to an alpha keto acid. Here's another example here. Here we have an amino acid, and again, it's this group here that's going to be transferred to the alpha ketoglutarate, and what we end up with then is this being converted to an alpha keto acid, and this results in another amino acid. So amino acid number 2, we call it. And in this case, what we have is glutamate. It turns out that the amino acid alanine is converted to pyruvate by transamination. So the enzyme is ALT, or alanine aminotransferase, and this is found in the liver. So when they find high levels of ALT in the blood, this indicates liver damage because the liver has allowed the ALT to leak into the bloodstream. Now, it turns out the transamination reactions are reversible, and they're dependent on the concentration of reactants. The reaction can proceed in either direction. So the concentrations are going to be regulated by keeping breakdown and synthesis in balance. And you know, we can see here, for example, if we have too much of this, then the reaction will shift this way, okay? But if we have too much of this, it will shift this way. So that it keeps breakdown and synthesis in balance. So the reaction of pyruvate and glutamate, as I said before, is how alanine is synthesized in the body. So that was transamination. Now let's talk about another reaction. So the glutamate that's formed from transamination is what we call an amino group carrier. So it provides the amino groups for synthesis of new amino acids. So we're going to talk now about oxidative deamination. And this is the conversion of an amino acid, so this amino group, to an alpha keto group with the removal of ammonium ion. So here we have glutamate. Again, this was formed in the transamination reaction. Notice here we have NAD and NADPH. And so we start with the glutamate. And again, this is also enzyme catalyzed. And what will happen is an ammonium ion is removed. Okay. So we have this, the NH3, and a hydrogen, okay, to form that ammonium ion, and then we're left with the ammonium ion and alpha ketoglutarate. What happens to the ammonium ion? It proceeds to what we call the urea cycle. The alpha ketoglutarate that is formed is then recycled for another transamination reaction. So what is the urea cycle? Well, it turns out that that ammonium ion can be very toxic to us. So the urea cycle converts the ammonium ion to non-toxic urea. So this is the structure here for urea. Notice we have the two amino groups, one here and one here, that are bonded to carbonyl carbon. So we have this carbonyl group here. So this takes place in the liver, and the urea then is transported to the kidneys and then transferred to the urine where it is excreted. So let's take a look here at what happens. So before going into the urea cycle, what we have is a reaction between ammonium ion and hydrogen carbonate ion. Also, the investment of ATP, and of course an enzyme. You don't have to memorize the enzymes. So in this reaction, this carbamoyl phosphate will form. Remember, the phosphate comes from the ATP. And this is the reactant in the first step of the urea cycle. The urea cycle has 4 steps. I think I can get it all up here. Okay. So here's what happens in the urea cycle. So in step 1, we have the carbamoyl group that's transferred from the carbamoyl phosphate, okay, to this orthanine. Okay, so this is the orthanine. The carbamoyl phosphate transfers this group here, okay, to the orthanine, forming this citrulline. So this is an exergonic reaction, and what it does is introduce the first urea nitrogen into urea cycle. Remember, urea has those 2 nitrogens. And we also see here the conversion of ADP to ATP in this first step. So in the second step, this citrulline here will combine with aspartate, and this is what we call it. This is a protein amino acid, to give this molecule here argininosuccinate. And here we see an investment of ATP, okay, for this particular reaction. So then when we get to step 3, the argininosuccinate is split into arginine, again an amino acid, and fumarate. And we know that fumarate is one of the citric acid cycle's intermediates. So now in step 4, what we have is a hydrolysis reaction. So the carbon nitrogen bond of the arginine is hydrolyzed, and again, this is a catalyzed reaction to give urea plus the orthanine, which is the reactant for the first step of the cycle. So here is the urea. So urea and orthanine. The orthanine then goes into the cycle and goes through the 4 steps. And of course, then the urea is then transported to the kidneys and transferred to the urine. So what is the net result of the urea cycle? Here we have elimination as urea of the carbon from carbon dioxide. Nitrogen is from the ammonium ion, and then there's also a nitrogen from aspartate. So the 2 nitrogens in urea come from different sources. One is from the ammonium ion, and the other one is from aspartate. There's 4 phosphate bonds that are broken in order to provide energy, and there's the production of fumarate, which is a citric acid cycle intermediate. There are many hereditary diseases that are associated with defects in the enzymes for each step of the urea cycle. So, for example, one of them is hyperammonemia. As I said before, ammonia is highly toxic to living things. And actually, fish are able to eliminate ammonia through their gills, but we can't do that, which is why we have the urea cycle to convert the ammonia to the non-toxic urea. But anyway, in this disease, there are abnormally high levels of ammonia in the blood. And this is associated with an enzyme defect in the urea cycle. This is a disease that causes vomiting in babies, weakness, lethargy, irregular muscle coordination, and also mental retardation. How's this treated? Well, transfusions, dialysis of the blood, and also the ammonia can be chemically removed. So, as far as long-term treatment goes, a low-protein diet is very important, as well as eating very small, frequent meals. So, let's talk a little bit about the fate of the carbon skeleton. There are three common fates of the carbon skeletons of amino acids. They can be converted to pyruvate, to acetyl coenzyme A, or they can be converted to an intermediate in the citric acid cycle. The carbon skeletons can be utilized to generate energy by passing through the citric acid cycle into gluconeogenesis, which is, if you remember, the pathway to form glucose, and by entering the ketogenesis pathway in order to form ketone bodies. So the two types of amino acids, we have the ketogenic amino acids, and these are the amino acids that are converted to acetyl coenzyme A or acetyl coenzyme A, and they enter ketogenesis. The glucogenic amino acids are the ones that can proceed by way of oxaloacetate to the gluconeogenesis pathway. Both ketogenic and glucogenic amino acids are able to enter the fatty acid biosynthesis by means of acetyl coenzyme A. So here is just a little chart that shows this. So we have alanine, cysteine, glycine, serine, threonine, tryptophan. Okay, they can be converted to pyruvate. The isoleucine, leucine, threonine, tryptophan to acetyl coenzyme A. Just take a look at this. Remember, the acetyl coenzyme A goes into the citric acid cycle. Here, the alpha ketoglutarate, okay, that could be from arginine, glutamic acid, glutamine, histidine, proline, okay. Succinyl coenzyme A, that could be isoleucine, methionine, threonine, valine, the fumarate from phenylalanine, tyrosine, and oxaloacetate from asparagine and aspartic acid. So just take a look at this. You can see to get an idea of the fate of those carbon skeletons from the amino acids. Humans synthesize about 11 of the 20 amino acids, some say 10. Remember, the non-essential amino acids are the ones that are synthesized in the body, and the essential amino acids are ones that cannot be synthesized by the body, and they must be obtained in the diet. Essential amino acids are synthesized by plants, and meat contains all of the essential amino acids that we require. Non-essential amino acids will derive their amino groups from glutamate, and glutamate, as we know, provides nitrogen for the synthesis of other compounds that contain nitrogen. So how about purines and pyrimidines? These are the bases, remember, that are part of DNA and RNA. So intermediates in the non-essential amino acids, pyruvate, oxaloacetate, alpha ketoglutarate, 3-phosphoglycerate. So we already saw that in the diagram that I showed you before. I just wanted to give you another example. Here we have phenylalanine, and this is an essential amino acid. So this is one that we have to obtained from our diet, and it's converted to tyrosine. Well, there's a disease called phenylketonuria, PKU, and this is where phenylalanine is not converted to tyrosine. So what happens is there'll be high concentrations of phenylalanine and phenylpyruvate in the blood and also in the urine. So if this disease is not diagnosed, what will happen is it will cause mental retardation by the age of 2 months. So a lot of these babies were born with a lot of problems. Many of them were institutionalized by the time they were 2 or 3 years old. And anyway, since the 1960s, the hospitals routinely screen for PKU. So what is the treatment for something like this? Well, they need diets that are low in phenylalanine. And so there are special formulas that are made for these infants. And as they grow, then usually they have to eliminate meats from their diet because, again, the meats contain all of the essential amino acids that we need. And they usually use low protein grain products for their diet. Anyway, we briefly went over protein metabolism, and this is the end of the module 12, and also the end of the course. So hang in there. There's only another week or so left. And if you have any questions, as usual, come and see me or email me.